Librarian here at the Medical Center Library here at the University of Kentucky, March 29th, 2023. And we're about to start one of our research workshop series presentations. Today's session is Data Management Planning for Systematic Reviews. And I will let Lauren Robinson and Rebecca Morgan take it from here. Thank you.
All right. Hello, everybody. My name is Rebecca Morgan. I'm one of the MCL librarians. I share covering health sciences, and I also cover the College of Pharmacy. Lauren, do you want to briefly introduce yourself?
Of course. Hi, everyone. My name is Lauren Robinson. I am the Medicine Liaison Librarian. I serve the College of Medicine.
And today we are going to go over data management planning for systematic reviews. And in today's session, we are going to cover a few tools that might help you in this process. SearchRxiv, yes, that is actually how you pronounce SearchRxiv. I always forget that, it's confusing. We're also going to talk about how to use Endnote in the process of maintaining your citations as you're going through or even preparing for the inclusion and exclusion process. Code books for systematic reviews, and we're going to talk about the importance of building or having a protocol for your systematic review and how that can help you plan your data management strategy ahead of time. But before we go into that, we're going to talk a little bit about why managing your data is actually important for systematic reviews. I think when a lot of people think about data management, they're thinking, oh, it's for my grant, it's for my research project. Do I really need to do it for a systematic review? And well, yes, because systematic reviews and other evidence syntheses such as scoping reviews, umbrella reviews, any type of these advanced reviews are at their core about analyzing data from data. So you're not only pulling data from other research projects and analyzing that. In the process of doing that, you're also creating a lot of your own data. And for the systematic review process to be truly well done, you need to make sure that you are managing the data that you are creating. And so what types of data should you be managing while you are doing a systematic review? Because the concept of data is kind of amorphous. Like, well, what is data? It can mean many things. So the things that you should always be documenting and tracking are even simple things, just like what databases did you use, on what platforms, what indices did you search, how were your searches completed, how is your hand searching done? Like, did you go through certain articles? Like, you came across a systematic review in your search results, did you automatically go through the references of every single one of those? So processes like that, they need to be documented. So it's not even just the numerical data, it's also tracking your methodology and how you actually completed your work. You need to track the dates the searches were done because new articles are added to databases every single day. So you need to say that this was done at this point. So if someone comes back and says, oh, but this article was published in October, you can always point to the fact, well, mine was done in September. The peer review data, the total number of records, the automation data, how your data was deduplicated, all of this stuff needs to be tracked because there's an expectation that this information will be reported out. If you follow certain guidelines like the Prisma S, which is just for searching, or even Prisma, or even the Cochrane guidelines, they all have a certain expectation of what information is reported out to make sure that your search is one, able to be looked at and said, okay, this was done properly, but also to make sure that it's reproducible. And reproducibility is really key in almost every kind of systematic review, any process like this, because you want to be able to show what you did and have other people to be able to go back and check your results by duplicating your search and duplicating your methodology. And it is so important to do all of this because there are major, major, major problems in systematic reviews. I think many people just assume, oh, I found a systematic review on this topic. It's the gold standard. It must have been done well. I'm going to assume it's a high quality article, and I'm going to change my clinical practice or change my handling of situation X based on what the systematic review is saying. And that's actually increasingly problematic because everybody's doing systematic reviews because they're like the hot thing to be done. And that also means that there's a deluge of them and they're not being done properly. And a big part of that is because the data is not being tracked properly. In this very scathing critique in the European Journal of Pain, they found and I will just go ahead and read this quote, available evidence indicates that almost all systematic reviews in the published literature contain fatal flaws likely to make their conclusions incorrect and misleading. Only 3 in 100 systematic reviews are deemed to have adequate methods and be clinically useful. We argue that the results of most systematic reviews should be dismissed. Forensically critical systematic reviews are essential tools to improve the quality of trials and should be encouraged and protected. And a big part of this is because if you're not doing the process right, if you're not doing a truly accurate search, if you're not documenting how your search is done and how your results are found, you could be adding bias to your search. You could not be truly finding the correct results. You're not even interpreting them wrong. Even from the beginning of when you're starting the process, it can be flawed from the beginning. And as you can see that going back and they did a very thorough examination of systematic reviews, they found only 3 in 100 were actually deemed to be adequate methodologically and to be clinically useful. And that should raise alarm bells to everybody. And so some of the things we're going to talk today will hopefully help correct some of these issues, or at least make you think about how you're handling your systematic reviews in the future. And I just covered that. And we're going to help you figure out how you can be better prepared. So as I alluded to earlier, and Lauren, please jump in if I gloss over anything that you think needs to be covered, reporting guidelines have become much more precise. I had mentioned the PRISMA checklist, which is a very, very, very detailed checklist that points out every single thing that you should be documenting in your systematic review and how you get to those points and how you find that data or in those reporting points. You know, that's up to you. You can use various methods, various tools to find this information or to generate this data, but ultimately you find it by tracking what you're doing, understanding the guidelines and what they're actually asking, and documenting, writing down everything you're doing, because this is a long drawn out process. There can be interruptions, and 3 months from now, if you've had a stop in what you're doing, you need to be able to go back and pick up where you were. I'm not going to go into the actual ins and outs of the PRISMA checklist or anything like that today. We just don't have the time, but we do have another research workshop series, an earlier one on the PRISMA checklist. If you want to learn more about that, if you want to learn more about those reporting guidelines for systematic reviews, I encourage you to visit our research workshop page, our guide on our website, on the Medical Center Library website, and a reporting is available for you there.
I will put I'll put that in the chat if you are interested directly to the YouTube video on the PRISMA checklist. And I will also add that, like, there's multiple PRISMA checklists and you have to do all of those to meet the standards for reporting. So there's, like, most people just say we use this one checklist. So I really recommend checking out that video, like Becky said, because we talk about the various checklists and what they really mean.
And we're going to move now into talking about some specific tools that you can use to manage your data when doing a systematic review. And Lauren, you're going to start us off with SearchRxiv. Let me just get to the page, please.
Can I do the slides first, though?
Oh, I'm sorry. Yes,
no, it's all good. Sorry. I'm having some Zoom issues today, everyone. Just muddling through with sharing slides.
Sorry. Are you seeing it? Oh, Lauren froze.
Oh, no, it's all good.
Okay.
So on slide 11, do you mind going on to those slides for me, Becky? I might have frozen.
You did freeze.
I'm sorry. Okay, now I'm here. So A little bit about a search archive. So evidence syntheses, we've already talked a little bit about they can take years and years to complete. I will say I was just writing methods up for one I started in 2020 and we've done updates on because it was really large. And I wish I did better data management when I first started these type of reviews. I spent a long time trying to figure out what I did. Now I'm much better at that. So we kind of cover these things that you need to like document all this information. And there is a link to Prisma S over here on Prisma Searching, and it includes all the requirements. If you want to learn more about that, again, we have also the link that we provided just a moment ago. So on the next slide, I want to talk a little bit about why this is important to SearchRxiv. So there's a movement in systematic reviews right now that is indicating that we need to make our searches available. It is now a requirement on the checklist to make our searches available. And SearchRxiv has been a product that was developed to help with that. So what it does is it allows you to submit your searches, and each search gets a unique identifier, gets a DOI. So just like your paper or your protocol gets a DOI, your searches get a DOI. And this is really important for the validity of our searches. It's important for the transparency and reproducibility of systematic reviews. This is an interdisciplinary tool. It's actually produced by CABI that does a lot of agriculture things and a lot of other science related things. It is social sciences, it's medicine, it's everything in between, including agriculture. They're trying to actually build, and they're requesting more and more people outside of medicine to put stuff inside of our SearchRxiv, because medicine and healthcare is kind of where these things rapidly come out right now. So there's just a lot more of them. So when you're in SearchRxiv, which slide are we on, Becky? I'm sorry, I think it's frozen on me. Oh, okay. There we go. So, so it will support you as researchers for storing and sharing searches from evidence syntheses. Like I said, it's transdisciplinary. It supports open access research, which is really great because the goal of these is to have research be more transparent and synthesize all the stuff from systematic reviews. So, you can go on to the next slide, please. So again, you can post your searches, obtain digital object identifiers, DOIs, link searches to published articles as relevant, find it easier to follow best practices for structuring the search strategies, and easily find relevant searches. So this is what's really cool about this is if you're not sure where to start, you could go search this and see how other people are searching, which is one of the hardest things sometimes for people to do, is to know how to start searching for these topics, especially topics like activities of daily living. I have a string that's probably like 2,000 characters long for activities of daily living. So just like this can be a really good tool for that as well. So can you go on and click the link for me, Becky? I'll do a little demonstration of it. So this is what SearchRxiv looks like. You can search topic. One other unique thing about SearchRxiv is that they've come up with standardized metadata. And for the non-librarians, it's data about data, and it's data specifically to help you retrieve inside of these databases. So what you can do is that when you submit a data set, so a search to SearchRxiv, you will fill out a standardized metadata, and that will make it more searchable. So if you have questions about this and you're wanting to submit, we highly encourage it. Some of our librarians are starting to do this more and more. Please reach out to us. We can help you through the process to make sure everything is standardized properly. And I can show you what one of these look like. Can you just click on one of the ones on the bottom right there, some of the most latest searches? So this is hey, look, this is actually some of our librarians and some University of Kentucky faculty here. There's Kayla and Jason are both on this call. This is one of their searches. So it is a public policy, I believe, and an environmental intervention search that they've been working on. And you can see here the search string. So this is the one I believe, I can't tell which database this is. I have too many monitors. So you can see that this is from
CAB abstracts and
CAB abstracts. Okay, so Web of Science CAB abstracts. And I was just going to CABI in my brain there, but you can see the number of results that the data was exported. the search field. So this is all that metadata I was talking about. It's what makes it unique and easily findable inside of this. And you can actually link your protocol. So if you published a protocol or registered it in Prospero, or if you've done it in an open science framework, you can link all of these things together. And that's what's really, really nice. The goal of this is to have your publication, your protocols, and all your data linked so it's all reproducible and transparent. So this is the really powerful thing about SearchRxiv. And I really, really recommend it. I've been trying to get some of my researchers to put their searches in here. But yeah, so that's all I have about SearchRxiv.
Remember to unmute myself. All right, so now I'm going to talk a little bit about how to use Endnote in your data management for your systematic reviews. One of the reasons we're going to focus on Endnote is because it is freely available, provided by University of Kentucky, and is a very commonly used tool, at least to initially collect your citations from your various databases, and then also to run deduplication. There are big pros and cons to Endnote that I just want to cover always whenever I talk about Endnote. First of all, it's great because it's free through UK. It's widely used, so there's a lot of support for it throughout the university. It has a lot of built-in tools that are very helpful in the systematic review, or even just like writing process, such as deduplicating citations. You're allowed to group your citations. You can cite while you write and format your bibliography as you are actually writing your paper in Word. And you can automatically full text import if you link Endnote to our library catalog. There are desktop online versions. It's available for Windows. It's available for Mac. You can have shareable libraries. All that's great. The cons are, though, that it does not play well with the cloud. You can store your libraries in the cloud so you can work from them from different locations, but I beg you, do not open them and work off them in the cloud. Sometimes it works, sometimes it is disastrous and you can lose everything you worked on. And so one of the things that we're going to talk about is creating archive files and backups of your Endnote libraries, just to make sure your work is saved. Because one of the things that it has historically done, if you work out of the cloud too often, whether this be Box, whether this be OneDrive, even though it functions a little better in OneDrive, still I wouldn't risk it, is it can just sync and sync and sync and sync and think that you are just constantly downloading your references, and you can end up with like 5,000 duplications of your library. And on top of that, sometimes it just corrupts and you just can't open it, and then you can't even work with it. So always want to warn people, please be very, very wary of working off the cloud. If you absolutely have to for some reason, I move around to too many different locations, just always make sure you keep a backup of your work that is dated, so you know I can go back to the most recent version and I will cover that. Also, and we will cover this as well, the deduplication tool is not perfect. You can't just run the deduplication and assume that you're good to go. And it's also just not the most intuitive software, but you know, the more you use it, the better it can be. So I will now try and jump into Endnote, and let's see how well this works. Lauren, is there anything you want to jump in and say before I move forward?
Yeah, I would say I put the OneDrive I guess I call it client, but like the OneDrive thing you can download. And one way I get around that, I do store everything on the cloud, but I have the client kind of installed on all of my computers. So there's like local copies that then are also going up to the cloud. And I've never had a corruption issue doing that. So I work on 2 or 3 different devices, and this does allow you to do that. So that's one way around it. There is a chance for corruption always, but I have not run into that. And that is one way, because there is that local save that then just gets backed up to the cloud. So that's one workaround for it.
Yes, I use that a lot too. I think that is very useful, and because OneDrive is provided for us at UK, again, that's a very easy tool to use. Still, I really encourage you to use backups, because if you are jumping from too many different computers, or if you're copying and pasting it from one place to another, and Endnote files just corrupt very easily. It is kind of a shaky software.
Right. I always save a compressed file of all of mine. So like it's a compressed saved one in a safe place.
And I probably do too many and end up with way too many different saved libraries, but you know what? Lots of copies keep stuff safe. That is the old acronym LOCKS that we always say for backing stuff up. So just to test, are you seeing my EndNote library, just an empty one? Okay, so this is just a completely empty EndNote library. And I also just want to give the caveat, I am not going to go very much into like using the basics of EndNote. We're going to operate on the assumption that you at least have, you know, a rough understanding or at least an understanding of how to export your citations into a file that you can then put into EndNote. So from your database, you know how to export into the citation manager from PubMed, create a file, and then we are going to now add them into a library. And if you are not that familiar with EndNote, if you need some help with it, we have a whole ton of EndNote videos and EndNote guides for you on our website. Some of them are just like little short tidbit videos. Some of them are very long, detailed introductions that are, you know, like an hour long, 20 minutes long to actually walk you through a more detailed process. So if you have any problems with EndNote, please, once again, visit our research guides. We have a whole one dedicated to EndNote, and that can hopefully clear up any problems that you have. But just for the sake of time, we don't have time to delve into too much.
And I put that in the chat as well. I put that link in the chat as well.
So the main thing that I think is absolutely critical when you're doing a systematic review and you're using EndNote and you had your searches done and you're importing your results is that you use the grouping function. And if you look on the left side here, can you see my cursor moving? Okay, under my groups, if I just right click on this, I can create a group. And what you should do is you should always create a group for every database that you search. And I also take the extra step of putting the date that was searched just to make sure this information is captured multiple places. So I'm going to go ahead and I'm going to create one for each database really quickly. And some of this is going to seem so simple because it is, but it's really the point of just taking these simple steps will absolutely help you in the long run. So now that I have my group set, I'm going to go through and import. And so I already have the file saved. I'm not actually going to go into the databases. And I'm going to start with PubMed. And so my PubMed files, and I just kind of got some random ones on a topic. I've imported. Now that they're in imported references, all I'm going to do is select all of them and just drop them into the PubMed group. And now I know that as part of my search, I had 10 results from PubMed. I'm going to replicate the same for the other ones really quickly. These 500 are from Embase. So I'm going to go through. Once again, I'm going to select all of them and I'm going to drop them into Embase. And as you can see, it's tracking that I have 510 total references at the top. I have 500 in Embase, I have 10 in PubMed. And again, let's just do Web of Science really quickly. They're importing. And once again, I'm going to select all of the imported references. So they're all highlighted. I'm going to drop them into Web of Science. So right here, we already have outlined one of the first things that you need to report according to Prisma and most reporting guidelines. How many total references do you have? 986. And how many in each database? So I have this all right here. You want to save this library and not touch it. You always want to have a backup of your pure search results that are completely untouched, because if something goes wrong in the deduplication process, if multiple people are working in the same EndNote file and something goes wrong, you always want to have a clean copy to go back to. So I always stop right here and save the library and call it like clean copy with the date, imported references, and then I'm always able to work off it. And then what you can do when you actually want to do your deduplication is you can create a copy, you can save a copy of it, and then just rename that copy something like deduplicated systematic review results with the date, because you want to track like when you're working on it. and you start working out of that one. So when you're doing deduplication in Endnote, another thing that you want to really be aware of, and this is why you need to save that first library with the pure results, because as you start going through deduplication, and of course no duplicates, I can't believe that actually happened. As you're going through the deduplication process, it's going to just start removing them from, I can't believe no duplicates are found. You're just going to go through and it's going to start changing the numbers here as you remove duplicates. And so as you, let's just say I'm deduplicating, I'm not just like deleting them randomly, but as you go through and you're selecting, I want this one, I don't want this one, I want this one, not this one, like, unless if you were keeping track with every single one, what database it's coming out of, you're not going to know, but you are going to see it start to change in these groups. So when you complete the deduplication process, you will once again have the second thing that you're supposed to be reporting according to Prisma, and that is how many duplicates were removed, and then what the adjusted numbers are for each database. It is not enough just to say, I took out 300 duplicates. You need to say, I took out 25 from PubMed, I took out 100 from Web of Science, and I took out 75 from Embase. And
we have a question in the
sure.
chat. Can you show how to do the deduplication again?
Oh, sure. So you go under library and find duplicates. And what normally happens, and I should have checked this to make sure I was going to get duplicates, is you get pop-ups. You get a list of pop-ups. It'll start, you'll get a new thing under here on the left that says duplicates. And then you'll get pop-ups of two that they think are the same, and you'll look at them and you'll compare all the data in the record next to each other and select which one you want to keep.
Hey,
Lauren,
Becky.
you're about to say something.
Yeah, could you just like double import PubMed? Like just
Oh, yeah,
import
that's
the
actually
PubMed
really
thing
good
again,
idea.
and we can show you how that looks. I will also throw about the deduplication. I recommend really considering like what you're deduping. There are settings, and I think we can show you those too. There are settings. Endnote is very, very sensitive in deduplication. There are other tools, and we talk about those in some of our other sessions, like SR Accelerator for deduplication and things like that. So this is one way to be to dedupe, but there are like, it's pretty sensitive. It has to be the exact same name. For example, if you use initials versus the full name, it's not going to count that as a duplicate.
Yes.
So it's not perfect. We recommend using multiple tools for deduplication, but we do have sessions on that as well.
And there are multiple strategies, and ultimately what you need to find is just the deduplication process that you are more comfortable with. I will cover doing a fuller version in Endnote for this, but yes, there are other tools you can use. But even so, you still almost always need to start in Endnote so you can create the file that you then import into these other programs. So still creating that clean copy from Endnote where you have just your pure results that are untouched is really critical as a backup file that you need to have for your systematic reviews. So again, just to demonstrate the actual deduplication, I'm going to click library, find duplicates. I imported the other ones in.
We also have a question, another question.
Sure.
Yeah, while Becky's doing that, I can answer it.
Oh, okay.
So how do you decide which references to keep if it's duplicated in both PubMed and Embase? So it's really like your own expertise as librarians. Like I think most of us probably keep either PubMed or probably PubMed ones the most often because they are usually the most full record. It's just a much more like comprehensively indexed database than like CINAHL would be, or Embase is pretty comprehensively indexed as well. But there is no right or wrong. It's just, I usually keep PubMed because they have the PubMed IDs. They have more unique identifiers that it's easier to manage later, but there is no right or wrong answer to that.
Are you able to see the find duplicates?
Yes.
Thing? Okay, so you're seeing the library with the find duplicates above it?
Yep.
Okay, so once you hit find duplicates, this is essentially what happens. It gives you two options. Now, these were from the same database, so there's not much difference. It was able to check it, but basically you look at the author names, you look at the year. It can be really helpful if it's included to look at the DOI, because if that's identical, it's obviously the same article. And then you say, okay, I want to keep this one. And then it goes to the next one. And then I want to keep this one. And if you're not sure, you do have the option to skip it and keep moving through the process until you finish all of them. If you have to stop, it's no problem. You can just close it out. And then this duplicate references is just going to stay here on the left until you finish resolving all of them. And you can just click back into it, and it should, if my computer wasn't freezing, or I guess you can just go to library, find duplicates. It will just find the duplicates again where you left off. All the ones you resolved, it will have moved. The one that you did not select into trash, and the one that you did select to keep will stay in the group that you put it in. As Lauren said, the deduplicator is not perfect. I mean, especially when it's coming across different databases, because databases index the records differently. So if one has the full first name included of the author and one has the initials, it might not recognize that. Sometimes there are abbreviations in the titles, or it'll be the English version of a German or of a non-English language journal title, and that will also confuse it. So if you are deduplicating an EndNote, you run the deduplication process, but then always go through the results by hand at least one, I would say at least 2 other ways. And the ways you can do that are by sorting by author and by title. So if I sort by author, either, you know, starting with A or Z, you can go back. I'm going to get off duplications and just go by author, and you can go through and start saying, okay, here's this name, looks the same. Oh wait, it's the same year, it's the same title, and then I'm just going to manually delete one. And then you do this after you run through all of that, every single one of them, and it can be fairly fast because, you know, your eye does jump out and catch the 2 that look the same next to each other. Then you can go to title and do the same thing. Instead of focusing on the author, you focus on the title, and then you check, oh, it's the same journal, oh, it's the same author, it's the same record, and then delete it manually. By the time you get to the second or third way, you will definitely only have a few, but you'll be surprised how many you still find that late in the process that are actual duplicates.
Yeah.
So again, because the EndNote process is not perfect, I highly recommend you scanning multiple ways, including at least twice manually, using one of these options, author or title. Lauren, were you about to say something?
Yeah, so like, and I tend to, like, I don't do as much manual searching as like Becky does. I tend to run it in multiple different tools. And if you go up to file, I think it's file preferences, I believe, you can actually change, or no, it's under edit. I'm sorry. Preferences. The one way I do it is under duplicates, you can actually change that. So sometimes I'll get rid of authors and I'll just run duplicates on year and title. And because I want to just see which ones are exactly the same in year and title, because the authors could be different. I'll run multiple different types of deduplication and go through so it's still kind of a manual search, but it's, instead of just going through the entire list, I'll run it with different things that I'm trying to exactly match. Sometimes I can run it into another deduplicator like SearchRxiv's deduplicator, and I'll do multiple things. So there's different ways to do it. You can manually do it if that's your preference. I tend to just like use different sensitivity methods for doing deduplication. And my method doesn't always catch every single one. Sometimes you as the researcher would have to catch like maybe 1 or 2 that I missed. But there are other ways as well. But it's like Becky said, it's really important to make sure you're doing different types of deduplication. And it's all going to involve some manual deduplication. It's just there are tools that can help you with that as well.
And again, it's really just coming down to you identifying the preference that works best for you and the system that works best for you. There's not a right way to do this. There's not a wrong way to do this. There's just a right way to use the tools, or, you know, a better way to use the tools, not even a right or wrong way. So I encourage you to explore looking at the deduplication tools that, you know, we've talked about again in other research workshop series. And exploring them, because you might really find that one is just way much more up your alley, intuitive and useful for you. But in terms of EndNote, again, they do have the built-in deduplicator. That's great. You're still going to be using EndNote because even if you're using SR-Accelerator, you're still going to need to take your files and export it as a .xml or a .ris, I can't remember which it has to be, in order to import it into one of these other tools. So you're still going to be using EndNote. So you still need to be able to create these clean files that you're saving different versions of, or clean libraries. So one with just your imports, one maybe after you ran deduplicator in EndNote, and then one that is your export file, or that is going to be what you export back in from SR-Accelerator. And an easy way to do versioning that doesn't create these confusing libraries where it's, oh, which one was I working in? One, make sure you're following good data management practice and naming the files so you know exactly where you were, like putting the date in it. But also, you can create these compressed libraries, these .enlxs, that will just basically, they're smaller, they're like a zip file, and they will just be backups. And so if you ever need to go back and find another version, just find the last enlx file, click it open, and it'll just pop open your library in the form that it was when you saved it. So you need to just be very careful, document what you're doing, document where you left off. So if something happens, you know exactly where in the process to go back to. But like most things, I truly believe the key to EndNote is just saving versions. After you've done a major change, after something big has happened, just make sure that you're saving a copy of the library so you're able to go back and not only track your work in real time, like, oh, I made these changes, but also to be able to say, okay, if something goes wrong, if a file gets corrupted, I'm safe, I've protected myself and am able to move on. Is there anything else you think that I need to cover in EndNote, Lauren? Okay. So let me go back to our, give me a second, please. Let me go back to our presentation, if you don't mind. Are you seeing the last slide we were on?
Just saying is, oh, yep, now we are.
Now you are, okay.
Next slide, and we should be good.
Oh, that's just saying what.
Oh, okay.
That I had that
So,
extra slide.
so that you can see, like, you're going to have multiple EndNote files. You're going to have RIS files from your exported data from the different databases. I recommend saving those too. Like I create RIS, I save the RIS that gets imported into EndNote. I save all that data. So you can see when you start accumulating all this information, you have a bunch of different data sets. So let's talk a little bit about codebooks. You are probably familiar with codebooks if you do other research. Next slide, please. So we've actually been working on one for systematic reviews. You all might know that a codebook describes the content, structure, and layout of your data that you're collecting. It contains descriptions and structures and content for the layout of your data, and it's supposed to be self-explanatory and help reuse. I will say that we started talking about developing this and I started working on it because I have gone back to try and write methods up for searches I've done. And I'm like, what did I do? Like, where is this document? I know I saved it. What's the name of it? So I wanted to have a human readable, like codebook text file that could be a plain text file that could be opened up on any device and that would have all this content in it. So that's what you see. You see the top of this right here on the right side. If you can go on to the next slide, I believe, is that the only slide I have for codebooks? Oh, and I also wanted to show you this one. So you can see, like, this is one of my original searches. I have so many different EndNote libraries here, final dedupe, and this is exactly the one I was working on. I'm like, what did I do? What did I do? So, and you can see my newer systematic reviews, like on the right here, you can see it's much cleaner, right? I have my protocols, I have my search documentation, I have all these things. So I've really organized how I do this. And then in each one of those, I have a naming convention where I'm using the year. So where is this all documented? If you, as a researcher, were just wanting to look at this, you would not know what all of this exactly meant. So if I had this code book in here, you'd be able to figure out what all these different documents are and where things live. And it's a good thing for me when I need to come back to it.
Yeah, because oftentimes advanced reviews like these, especially if they're for a guideline, they're not dead documents that die when you complete them. There's going to be an update in 2 years or 3 years, and you're going to need to go back and know exactly what you did in order to not have to start completely from scratch and build it all over again. So
yeah, and this is a movement across the board. Some of the big players in systematic reviews, like the University of Michigan, are really promoting data management planning for this, like actually having a data management plan for your systematic reviews. And we're talking about that. I'm not saying like a huge data management plan that's separate like you do with a grant, but I think both this code book and what Becky's going to talk about next, the protocols, can really help you with that. Data management to make sure you're getting everything sorted for your reporting, what you have to report in order to be adherent with different guidelines. So we can show you the code book. Do you mind opening up the LibGuide for me?
no, that is no problem. Let me just find it. Oh my goodness, do I not have it open? Is it in chat?
It was, it's the systematic review guide. It's the third one over, getting started.
Oh,
There. Okay. So how to document your search. So I actually put it under documentation. So how to document your search. You scroll all the way down. We have a bunch of other tools here for documentation, but here's the code books. I also have the slides for this presentation here, but you'll get those as well in a follow-up. But the code book, it should download.
did it open it?
Yes, please.
Oh,
And it opens up in the browser. It is a text file because the goal of good data management is to use non-proprietary data formats. So I did use a plain text file here. So it's open in browsers. It's open. You can open up a notepad or whatever other tool you're using for plain text. It opens up in Word too. So what we have here is basic information about your evidence synthesis project, whether it's systematic review, scoping review. And we break it down into your general information, all the people who are working on it, if it's funded, sharing and access. Are you going to put limits on it? And there are, especially if you're working with international groups of researchers, there are certain like licensing issues. Copyright varies. In the US, we have a really broad copyright law that allows us to use things like covenants, and other countries don't have that. So just there might be some licensing restrictions involving abstracts and things like that. You can always reach out to a librarian if you have questions about that. And then there's also search method information. So this is the database names, the coverages that you're searching, the vendor, the dates the search were run, your Sentinel article. So what did you test your searches against in order to help the validity of your searches? What softwares did you use? The software citation, and what each investigator did, what their roles were, which ones did the deduplication, which tools did you use for deduplication, list your search strategies. And then I'm going to work on some of this data collection piece. So this is a living document. It'll be edited over time. We are going to put some information about if you use REDCap, how did you do extraction, and how did you maintain assurance? How did you norm? All of that would go under your data collection methods. And we're going to be fleshing that one out as we go, and it'll be updated on this guide. And then we also have the data file overview. So where are you storing your files? How are you going to be backing them up? Are there going to be regular backups? Are there versioning? OneDrive does do versioning. It does save so many versions behind, at least the UK version of OneDrive does. And there are backups of UK's OneDrive. So that's one reason I use that client that I put in the chat earlier, and that's where I save everything. One other option would be Open Science Framework here. And we talk about these in a lot of our other sessions. If you're working with people outside of UK, OneDrive's not a solution, but Open Science Framework would be, as well as your file name and your file structure. So I'll know what that protocol folder means. I'll know what my search documentation folder and what files live in there. So this is what's the really important part of making your data your data readable and understandable later. And this helps. These are all the things that I end up reporting often in a methods or in a protocol. And so it's really nice to have it in one place. So that's kind of what, so this is what the code book looks like for now.
just a second. Sorry, we have these transition periods.
Yes.
And now I'm going to talk a little bit about protocols. So if you're familiar with systematic review protocols, the purpose of them is basically to, I'm probably going to articulate this poorly, Lauren, so feel free to jump in if you want to. The purpose of a protocol is basically to pre-plan your work for your systematic review, to have a draft of your search, to have all your inclusion and exclusion standards thought out ahead of time, to have the purpose behind your search. Basically, you're creating a document that is not your systematic review, but it's going to outline absolutely everything that you are going to do in your systematic review. And the benefit of this is that it takes away the questions that you suddenly hit when you have all your articles, you have all your citations, and you're going through and you're saying, oh, wait, this is gray. Does this one fit in or does this one not? If you've already thought through in great detail your inclusion and exclusion criteria and you basically have it written out, it makes the process of actually going through managing your citations much more streamlined. Lauren, what were you going to say?
And I was just going to say this is a really great place to put your data management planning. And with these increased guidelines that we've talked about some here or there throughout this presentation, with Prisma and other things being more precise and launching newer versions that are more detailed for your reporting in these type of reviews, it's not always possible to put those in your methods of your final manuscript, right? Like your method section's fairly short in a review manuscript for a journal. So your protocol is a really great place to put all this and cite it in your manuscripts. And I put all of that information into my protocol. My data management strategy, what tools I'm using for data management, all that goes in the protocol, and then I can cite it in the manuscript. So then I'm still adhering to these guidelines.
And so you might be thinking, wow, protocol sounds great, but how the heck do I create one? Because, what do I know what to put into it? So we have created a protocol form, basically a protocol builder in DMP Tool. DMP Tool you might be familiar with. It's an open source tool used to create data management plans. Also a way to create forms that have guidance built into them. So we used it to create a protocol form that you can basically, and I will demonstrate it in just a second, that you can basically plug in your information and generate a PDF or a Word document out of it, and then you can put that into Open Science Framework or OSF. I mean, you could even put it, I guess, into UK Knowledge if you really wanted to, or as long as a DOI gets assigned to it, digital objects identifier, is that what it stands for? Yeah. As long as a specific identifier and a permanent link gets assigned to it, which it will in UK Knowledge or OSF or many of these repositories, then as Lauren said, instead of writing in all your methodology everything you did, you can just kind of do a brief overview and say, for more detailed work, this will permanently live, like the rest of the information is permanently saved in this protocol I created, and then there's the true outline of really everything that was done in your methodology. So you can access this plan. Again, we have a great systematic review guide for you. There's a section on creating a protocol that walks through some of this. And then there's this link for DMP Tool, the data management planning tool. Only thing you want to remember with DMP Tool is that, if you're going to follow this link directly, always be logged into it first. I know that's kind of annoying, but if you click on the link before you're logged into DMP Tool, it'll just take you to the login in DMP Tool, and you'll have to know how to navigate to it. So just to make sure that we have all bases covered, I'm going to show you how to do that right now. Oop. There we go. That's what I wanted to do. So you can just go to dmptool.org. You don't have to enter it from UK or anything like that from databases. And then we have I don't know if we want to say subscription. I don't know exactly what the term is. We have a relationship, we're an institutional member. Thank you, Lauren, of DMP Tool as University of Kentucky. So you can just put in your link blue with your email attached, continue, sign in with institution, and it'll take you to, oh, it didn't do it for me because I must have logged in recently. It might take you to your link blue login, and then it'll take you to the general entry page for DMP Tool. And what you do is you go up to create plan. And this is what you do for any DMP. So if you're creating one, if you want to use DMP Tool to follow like the NIH new data management sharing plans, you can do that here. But for the purposes of this, for creating a protocol, name the project that you're working on. I'm just going to call this test. If I knew how to spell test doc, and because this is mock, I want to make sure that's listed. It will default to University of Kentucky because you logged in with your University of Kentucky email. And then click on no funders associated, because when your protocol, it's not related to funding. It's not a grant based.
Yeah, and you can always go back to the guide we were just on and just click that link and not have to do this, and it will automatically populate it for you. So we do have that, and there's instructions on the guide there.
Sure.
And I put in, we have a whole like 40-minute presentation on this tool for protocol development, and I put that in the chat as well.
So if you click no funders associated with this plan and starting review, create plan, and now you're in the protocol building tool. You have to fill out some basic information, but basically it walks you through every aspect of what you need to fill in to have a protocol, from basic information like title, authors, if it's registered anywhere, to writing an introduction that includes your rationale and objectives, to the meat of it, which is your methodology. And in all of these, you will notice there's a section for you to enter the text, but in almost every case, we have provided a systematic review example, a scoping review example where applicable, and notes about tips and things that you want to remember. So there's example text of what you're looking for here. Not all of this is necessarily directly related to data management, but there are certainly parts of it that are critical to it. For example, writing down exactly what data items that you're looking to collect, obviously your search strategy and your information sources, but then also how you're going to complete your data synthesis. Which one has the tools under it? Does it list the software that you...
I think it's kind of housed in many areas. It's both in the data synthesis, it's in the search strategy, and information sources. It's kind of all that stuff lives throughout the protocol.
Please continue.
Oh no, I don't remember exactly where you list, you can list the tools in multiple places here, I think.
And it prompts you again with this guidance on every page what it's looking for, and so it kind of takes the questions out of what data should I be managing, because one, the protocol gives you a place for it and the guidance tells you exactly what it's looking for. And so this is a tool that you can use to prepare yourself and set you up for the best success possible when starting a systematic review, because it kind of, as a data management plan would for a grant or a research project, it kind of makes you forcefully think through what software am I using, who's going to be doing what information am I actually collecting, what is my inclusion and exclusion criteria. So you're going in with that clean mindset where you've already accounted for so much of the work that we've already talked about, saying I will use Endnote or I will use SR Accelerator to do my deduplication. I will then import it into RAN and use that to actually do my title and abstract level weeding of citations. This will very clearly define the steps that you will be taking, but it just takes a lot of guesswork out of the whole process. Lauren, what did you want to add to that?
Not at all. I didn't have any other things to add. I just saw that we only have 4 minutes left.
Oh, my goodness.
I wanted to see if there's any questions anyone had before. Becky and I could talk about this forever, I think. So I just wanted to see if there's any questions.
And did we answer Courtney's
Yes.
question well enough about deciding? Okay, because I was like struggling through getting the duplications to work.
Yeah, I think we did. You can really decide on either. And the one thing I will add to that one is you can see at the very bottom, when you have that 2-screen part of Endnote up, you can see at the bottom which database the record came from. It'll say like NLM or Elsevier or Embase or wherever. You'll see at the very bottom what platform it came
And I primarily choose the one that has the most information in it. If one has a DOI and the other one doesn't, if one has only partial information, like has the volume but not the issue, I choose the one that has everything. So really it's up to you which one you choose, as long as you're getting rid of the one that is one of them that is a duplicate.
Yep. Any other questions?
And of course, don't hesitate to reach out to us after this. We will have the slides with our information, our contact information, including it, and we should be able to help you there. And yes, please unmute and ask your question.
My question was, how much will the medical library be helping us with the systematic review search? Do you guys help us with the search, or like how much will you be able to do for us?
So we do have, let me try and find and pull up
I got it.
the systematic review page.
I got it. I can put it in the chat.
Okay. We will do however much or how little you would, let me share my screen again. You would like us to do, basically, as long as it meets certain conditions. So so we're willing to help from 3 different levels, from consultant to collaborator. Consultant's the simplest. Do you feel like you can handle this mostly on yourself, your group? Do you just need us to maybe like give a little expert advice, like, oh yeah, use this term, but other than that, you're doing good. Look over your search, make sure that things look good. Next, we're facilitator, and the difference with that is that we do require that we're at least acknowledged. It is acknowledged that a librarian assisted with the search in the final publication or the final output, whether it's a poster, whether it's a paper or what. And then we just do much more intensive support for you, where we actually advise on citation management, suggest tools and research, demonstrate how to use them, things like that. And the third, and I would say definitely most common way we are used, is as collaborator. And we do require authorship if we're going to help at that level, because we are essentially building the entire search for you and deduplicating everything for you. And we would do it with your work. I mean, we would come up with it together, but in terms of actually building the searches out and constructing them in the databases, identifying gray literature, conference proceedings, doing some of those much more complicated deep diving searching, and then also tracking the methodology of the search process, we would take that on and then share it with you to do the evidence synthesis. But because a systematic review cannot be done without the search, and it cannot be done well without a proper, well-formed search, we do require co-authorship, because the contribution that we would be providing is so in-depth and so large. So yes, we do different levels of consultation based on what your needs are, what your team's needs are. Please don't hesitate to reach out to us. Do we have the link to the? Yes.
Yep.
At the top, we have this link here, request a systematic review consult, and that will go just generally to our group, and we will figure out who is best capable for whatever time commitment or expertise in the subject area to help you. And we can go from there. Or if you know the person who is the liaison to your specific school or department, you can reach out to them directly as well.
Yeah. And I would say if you're thinking about one of these, reach out earlier rather than later. All of us are working on multiple of these type of projects. I often have a 2-3 month wait on mine, just because I am working on so many of them. Sometimes we don't have waits, but sometimes we do. So if you're just starting to think about this, reach out early. So we can definitely get you planned into our schedules if you're wanting to be, for us to be a collaborator with you.
Absolutely.
Another quick question was, when you're a collaborator, do you usually, like, what level of authorship do you get? Is it a middle author, or would it be like the first author?
Oh, No, just one of the authors thrown in the middle. It doesn't
yeah.
have to be any kind of special
Order or
signification.
anything.
Just as long as we are just included as someone who contributed as an author, it doesn't matter where we fall. And definitely we would not, I don't think any of us would feel comfortable saying that we would be even close to the primary investigator, because we would not take part in evidence synthesis. So that's the thing too. Most of us draw a pretty hard line at not being one of the people to select
Inclusion,
the actual
inclusion.
articles for inclusion or exclusion. Basically our job
Is
mostly
front-loaded.
stops
Yeah.
at the point of doing deduplication and handing you the citations to do the synthesis on.
And we do write the methods though, like we
And
do,
we'll write
people
methods.
come back to us and we'll write up all of our methods. We'll write up the protocol stuff and the data management pieces at that
Protocol
co-authorship
registered and all
level.
that.
Yeah, we'll help you get it registered. We'll answer your questions. Sometimes I even help people figure out which tool is best to do, like their title abstract review and their full text review. Like I'll show them, like these people use RedCap. Like we can advise you on that like part where you're doing your evidence syntheses, but we're not subject experts usually in your topic. So we don't feel comfortable like reviewing it, but we do a lot of front loading work with all the data management and searching, and then we'll write the methods at the end for you based off whatever journal you choose and their requirements.
Much.
No
Yeah, of
problem.
course.
Does anybody have any more questions for us? Okay, well, we are actually a few minutes over, so we should probably wrap this up. Again, don't hesitate